to come in. Till then, let's pass the mic around and hear who's in the audience. Can we get a mic and pass it around to the members in the audience and just tell us who you are, which company, what's your interest in this topic, maybe something like that. Start off with our friend over here. Yeah, there's a mic behind you. Yeah. Testing.
And I'm representing the DDR&D, the Directorate of Defense R&D, which is the Israeli DRDO. And Dr. Ari Gargir is one of our companies that we are trying to bring them to India.
Great, thanks.
I'm a neuroscientist. My interest is twofold. One, at a personal level, to see if I can change the quality of my blood, using one of these lovely gentlemen's technologies. The second is to, probably stand in line whenever they are commercial, to try and become their commercial partners and sell artificial blood.
Very good. You've got stretchers outside, so you can sign up there. Yeah, pass the mic behind you.
Sorry?
You've got stretchers outside. We can give you some blood. No, just kidding. Yeah. To the person behind you. Yeah.
Hi. So I'm also a biotechnologist. And apart from that, I work in venture capital. I know Dr. Ari, so I'm accompanying him, and I really love the technology that they're making. So that's why I'm here. And yeah.
Oh, excellent. Yeah, you've got a lot of fans over here. Yeah, pass it on to a young friend here.
Hi, I'm Nemesisa. I'm representing a family office, and also I run cross-border startup accelerator program. So very excited to know more about this technology, because health tech and biotech are as a sector, we are very keen on investing and also working with international startups who are doing revolutionary work.
Oh, very good. You got some money in the audience also. Yeah, let's pass it on to.
Hello. Am I
Yeah,
audible?
we can hear you.
Yeah. My name is Akanksha, and I represent my company OpenEye Partners. We help large organizations to engage with startups. So a lot of time, the corporates, our clients, they ask us questions regarding emerging technologies. So I'm here so that I can answer all their questions.
Excellent, thanks. So you can help connect these guys to
Absolutely.
the bigger firms, hospitals, and so on. Yes.
Pleasure.
Hi, I'm Dashmi. I'm from JETRO, the Japan Trade Organization. I just thought this was a very interesting topic, so I'm here to listen to what India and Israel are doing, so that Japan also is doing a lot of stuff in the medical field. So I just like to see how and where things are at the moment.
Very good. Japan has been mentioned as one country in this artificial blood space. We've got some more folks over here. Can we just get quick introductions from them, and then we'll get started. Microphone there.
Hi, it's Manik here. So I am from SBI Startup Branch. So we are working with startups in Bangalore. So
Excellent.
closely relating to all kind of startups, less than 10 years. So that's what we are doing.
Great. You got some startups here, right? So we can contact them also. Yeah. And pass the mic on to.
Hello, I'm Gaurav Ved, founding partner at Ventures Inc. I've been in the tech space. Now I advise startups on building products and innovation. I strongly believe that tech's going to be the platform on which everything that is more human-centric is going to be innovated and built on. So that's why I'm interested in health tech and the other vertical techs. What can tech do to solve real human problems? And this subject sounds interesting. I do believe that eventually human health is about blood and breath. If you can control those two, most people can be healthy, and so interested in this.
Yeah, thanks. And last, before we start.
Yeah, hi, I'm Vinay. I try to create content using tools that young innovators and startups can work on, insights that you thought leaders share. I'm looking forward to the session.
Very nice. Good. So we got a good full house now. There are more people in the audience than on the stage. So that's a very good sign. No, just joking. So we got a very interesting topic. For those of you who are not in the bio field, this may almost sound like science fiction. The coming blood revolution, the future of blood, so on and so forth. You've probably already heard about 3D printed organs that may be science fictionish to some extent, but now we're talking about really futuristic kind of stuff in terms of artificial blood. So can you synthetically, biologically create actual blood and then use that in so many different kinds of scenarios around the world? You all already know, of course, from real life, from the movies, from the news and so on, that there's a big shortage of blood. I know many of you may know of personal stories of people who needed blood but could not get the right blood at the right time in the right place, and they may have eventually passed away and so on. So hopefully there's a solution now. If you can create artificial blood, which is almost as good as the real blood, you can produce it in huge quantities, maybe at lower cost, put it in multiple locations. So anytime anyone needs blood, you can get the blood. So it can prevent loss of life, loss of quality of life. It can even be used to treat rare kinds of diseases and so on. So is this possible scientifically? Is this possible as a business? Is it possible as a global mass market? That's what we're going to find out here with a panel here today. I'm going to invite our two speakers very briefly to introduce themselves. We have with us Gargir, founder and CEO of RedC Biotech, and Praveen Kumar Vemula, Dean of Research, also a professor at the Institute for Stem Cell Science and Regenerative Medicine. So, Ari, tell us a bit about how you got into this field. What does your company do? What's it based on? And what excites you so much about being in this space? Ari, big round of applause, please, for our first speaker, Ari.
My background is biotechnology. I did a PhD in human microbiology, but I was most of my career I was in biotech companies, the pharmaceutical companies, bringing products to the market. But my interest in blood started many years ago when I was 25 years old, a student. And I had an accident. I had chest injury, and my life was saved by a blood transfusion. And at the time, I was a bit scared that I may get a disease from the blood transfusion. Nothing happened in the end, but it sparked an interest in the field. And only many years later, when I reached the Technion in Israel, India's IIT, and I learned about a technology that allows to cultivate stem cells in very large masses and produce any product. And suddenly I thought, this is how we could solve the big challenges of blood supply.
Fantastic. Thanks. Praveen, how did you get started in this? What's your backstory?
Okay, my name is Praveen Vemula. By training, I'm a chemist. I did my PhD in organic chemistry, and I run a bioengineering lab at InStem. So it's an academic lab, where we find an unmet clinical need and custom develop technology to solve them and take to the market through, practicing science entrepreneurship. That's the overall theme of our lab. And so far, we are like a field agnostic. Rather I would say we are a very unfocused lab what we run, because not one disease area what we focus, but as long as unmet need is big enough and there is a huge demand to solve the problem, I think that's where we pick the lab and pick the problem and solve them. And our story in coming into the blood area is actually literally started over a dinner conversation. So one of my friend, he was visiting from Germany, his entire PhD was characterizing the stored blood, how as a time-dependent manner, what happens to the quality of the blood. That was his study. So, and it's a very phenomenal information. What he provided is how much the deuteration happened during the storage, and it's a completely time-dependent manner. So that just sparked my interest to see. Then we started digging more information about and identified that there's a huge scope to invent there, to technology to where you can increase the quality of the stored blood. So that's where we started working on and developing the technology.
Great. So we've got two experts from two countries over here. Where else is all this blood research kind of stuff happening? Which other countries' initiatives are you aware of? We heard about Japan very briefly, but what other countries are you familiar with?
Yeah, well, there are different ways of solving the blood shortage. There's the chemical way, producing alternatives to blood. So there are a few companies and government initiatives in this area. The Japanese group that was mentioned are an academic group, and they're actually using expired blood units in order to extract hemoglobin and repack it and kind of imitate the structure of a miniature red blood cell. So that's one way of solving it. The other way is producing red blood cells from stem cells. And for that, there are a few companies around the world using different technologies, different cells from different sources. One of them can be stem cells that are derived from blood transfusions, primary cells. Others use immortalized cells. We're using a technology that allows to use stem cells that are derived from a donor, a one-time donor, a one-time process where we make stem cells that could be prepared and propagated, and you could maintain a cell bank that can theoretically last forever and produce cells that could last or could be produced for all human for eternity, theoretically, of course.
And where is this kind of work happening? You mentioned earlier, I think the US and some other countries are active in this.
Yeah, so there's a company in the US, there's a group doing a clinical trial on an academic level in the UK. There are a couple of groups, there's one in Korea, and we're in Israel using the induced pluripotent stem cell technology.
Praveen, you worked abroad also, right? You studied abroad and researched abroad. Any of this blood stuff kind of work was happening elsewhere also?
No, I think the US also has like a very advanced stage where we have, I think they're making like, as I mentioned, mostly like the chemical alternatives, like, for example, they developed this technology, erythroma. So it's basically a synthetic, I wouldn't say synthetic. What they do is like, if you just go into a little technical, your entire blood function comes from a protein called hemoglobin. So where the protein capture the oxygen and it acts as a supplies to every tissue, and also it takes the nitric oxide and supplies back, right? So this entire transportation done by this protein, your blood is primarily consists of your red blood cells. You have protein packed inside and you have a membrane, right? So your blood types and all comes based on what kind of your skin membrane these cells have, right? So alternative approach what people are taking is like taking the inside protein from, say, expired blood. You extract that protein and packing them with a new skin. They are giving a synthetic new skin which doesn't have this different cell types and so on, so that like anyone can receive this blood, right? So that is like a tennis ball analogy. You have protein inside and you have a cover which is like a much more safer and easily acceptable for universal donors. So that's the technology what people are doing. Like, I think primarily US companies have tried, and they are into like some of them into clinical stage level, and many of them in like a preclinical, like where the animal studies are being done, but a lot more development needs to be done. Yeah.
So this is becoming a masterclass on blood, not just a panel discussion. Tell us a bit about the ecosystem of blood now, like the market of blood. How much does blood cost? Where does it come from? How many liters of blood are out there in supplies? Any numbers you can throw onto this whole thing?
Yeah. So blood is a complex material. The only source today is from humans, from human donors, and we rely on their goodwill. So we're totally dependent on volunteers. About 120 million blood units are donated every year, but there's a calculated shortage of about 100 million blood units every year. So which means that we're actually supplying just a little bit more than a half of the blood that humanity really needs in order to save enough lives. Many people still suffer from inadequate or insufficient blood. In Western countries, the cost of a blood transfusion can reach about $200. I understand in India, the cost is less. The cost consists mostly of testing, because each blood unit has to be tested for 7 or 8 diseases before it's re-injected, and the blood type. The blood, has different types, the ABO, the RHD, which allows you to determine to who you're going to give it. And that consists the cost. The distribution of the blood types does not fit the actual market. because you have O-negative blood, which can fit all humans without the need to test. But only 6% of the population carry that blood type. So there's an imbalance between the type of blood that you could give everyone to that. So there's a constant shortage in O-negative blood. There's less of a shortage in O-positive blood. And all of the rest of the blood types are usually waiting on in storage to be administered. The shelf life, as Praveen said, is limited. And that all of these consist, the cost, logistics of the supply of blood are the main cost. But the most important thing is the human factor, because humans have holidays, have bad weather, and they're not already all the time available to come and donate blood. And we're going to solve that.
Yeah. Praveen, what's the cost of blood in India, and do we have enough blood? And what's the shelf life of blood and all those things?
So, for example, you can measure by the unit. Like each unit costs about like 1,700 rupees. So convert into like, let's say about $20 to $25 per unit. That is including the tested one. But like this is excluding of the hospital charges and operation charges. So, but still it's much more relatively a lot more affordable in India than compared to Western worlds. And regarding the storage, if you see like a typically human blood, once you collect it, you can store for maximum 42 days. Up to 42 days you can store them, and after that like it will be discarded. Although technically you can store them for 42 days, but it is advised that only up to 21 days stored blood can be actually transfused, because people done this time-dependent studies where after 21 days, if you see the quality of the blood declines, and if you transfuse the blood that's after 21 days, the stored blood, so that can cause a severe systemic side effects, and many times it can lead to like organ injury, and also it can happen. So, but unfortunately, if you see like most of the times when you go to the hospital, like you ask for the donor, they collect the fresh blood, but what the transfusion, you try to give maximum stored blood because you have to get rid of them, otherwise they cross the 42 days, you have to discard them. So that's why invariably like we end up having old stored blood transfused. So therefore I think there's a huge need in terms of like how to improve the quality of the existing donated blood. And regarding the need, if you see, I think you gave the global statistics, but if you think about India, right? So annually it's estimated that about 12 million units of the blood is required for the transfusion. And during like through various blood donations and this blood bank initiatives, like we get close to like 10 million units of the blood is collected. But like, despite of the scarcity, like every year in India, we discard almost like 0.5 million. That means 5 lakh liters of the blood is discarded because during the storage, the quality declines. So that's why there is a huge need is there, how we can develop innovative technologies to minimize your wastage and improve the quality.
Right. So our speakers have done a fantastic job of setting the context of why we desperately need artificially generated blood. So let me ask Ari now, how did you get this technology for the company, and how is it being used? Where are you in the whole life cycle, the pipeline of getting this blood out into the market?
Yeah, so the technology that we're using, what we're actually developing is based on 2 pillars. The first is the cultivation technology that came from the Technion, allows to grow the stem cells as single cells in suspension, which means that you could grow these cells very efficiently in a bioreactor. And it means that you could scale up indefinitely, not indefinitely, but as much as engineering can allow. And that means when you reach the scale, the cost of processing, the cost of harvesting and testing can decrease considerably, and also the cost of raw materials decrease significantly. Taking all of those factors, we can reduce the cost. On the other pillar is the stem cell technology. And we're using what's called pluripotent stem cells. Stem cells to begin with are very special cells because they can expand and grow indefinitely. Just in like in our body, our stem cells produce more and more cells for our lifespan. And the other point is that a stem cell can differentiate and turn into any cell type. It could turn into skin, into muscle, and also into blood. The ultimate stem cell is a fertilized egg that could produce a human with everything that he has. The stem cells that we produce are reprogrammed from a human sample. And the beauty is that we could select a human according to his characteristics. We could select a human who has universal blood, make a stem cell, make a master cell bank of stem cells out of this person's blood type, and this master cell bank is used afterwards to produce the blood for the people. We're going to produce blood that has specific nuances for the region that it has. So, for example, in Israel, we have a population which is Middle Eastern and European, and our universal blood will fit more, the nuances of the blood type will fit our population. And we're working with a government group here in India in order to do the same, to fit the blood types for the population in India and find the exact structure of blood that will fit the biggest population to minimize side effects and make it as safe as possible for people.
Wow. We'll come to partnerships a bit later, about your partnerships in India and so on. We'll come to that. Praveen, you're doing a whole bunch of bio things, right? Blood is one of them. So where are you in your blood journey in your organization? The kinds of synthetic blood, artificial blood products and so on.
Our approach is not like we are not making a synthetic blood and alternative blood, but our approach is like how to improve already donated blood, existing blood. As just mentioned, like, where your donated blood is so precious, like, but like we throw so much, discard so much because of the lack of technology to prevent the deterioration, right? So our idea is like if we can develop a new technology which can improve the quality while storing them, so we can solve this problem, huge impact we can make. So just to give any emphasis, like, what we have done is like, as I mentioned, like right now it's only 21 days stored blood is qualifiable for the transfusion, right? But what we have done is that we have developed a new blood bag. Now we can take this blood bag and we can, in connection with our currently practiced blood bag, you can have this a joint blood bag. So you can store in your regular bag. On 21 day, we can just pass to this new developed bag for 15 minutes and put it back. Then that is enough. What it does is like, now if you use this technology, even 42 days stored whole blood quality is as good as freshly collected donated blood. That is the level of quality we can improve. And in addition to that, like, we also shown that almost like a 25% of the shelf life also we can increase. When you use this technology, even 51 days stored blood can be transfused without having anyone. So, but just to caution that, like these all are demonstrated only in the animal studies. We are still haven't gone to human studies yet. So if you need more details about the technology background, I can give, but like the overall, what the approach, what we are taking is like, developing a new blood bag, which can enable to improve the quality. So that especially this would be very helpful, like when you have the rare blood too, right? Donations itself very rare, and throwing them or discarding them, like, because of the quality, deterioration is a crime. So that's why we thought like it will have a lot more impact having these new blood bags. Yeah.
Great, good to hear.
The fact that you have cultured red blood cells will have a longer shelf life.
Absolutely.
If you put together with that the blood bag that could extend the shelf life, and you have one blood type, the whole idea of inventories You won't have the dilemma of blood type, one type that has a longer shelf life. You're solving most of the logistic problems of blood supply.
Yeah.
So this will be a very additive, like in science, it's an additive process, but it will all sum up to maybe doubling the shelf life.
Good. Looks like a good collaboration here already. Let's turn now to the investment. Where's the money in all this space? We've got a few investors in the audience also. So how much money is involved in getting this into the market? Where are you in your funding journey? What kinds of funding can you turn to? Let's start with you.
Yeah, so stem cell development and specifically pharmaceutical development are costly. Until now, we raised about $4 million, a little bit more, but the investments will have to grow, going into preclinical testing and clinical testing, and in the end, scaling up into very large vessels. All of these will require large investments. Clinical testing is also a very big thing. And of course, the establishment of production facilities, those are in the tens of millions of dollars for each one. So the investments will grow, but as the risk will reduce in the development, we'll be able to meet and minimize the risk of those investments.
So Praveen, in your case, you need more research funding, you need more commercialization funding. What do you need?
So typically what we do is like with the conceptual level, for the technology development, we raise all the government funding. These are through various grants, Department of Biotechnology, Department of Science and Technology, and so on. We raise the money from the government. And once we develop the technology, then when we form spin-off company, for example, for our technology, we are just in the process of forming a spin-off company. Then we transfer the technology transfer will be done to the company. Then from the company, it's a completely private equity funding it's a completely dilutive funding what we generally generate and to take them to the market.
Okay. I've got a couple of more questions and I want to open up the floor to questions from the audience. My next question for you guys is, what are some challenges that are there in this field? What comes to my mind is public perceptions of synthetic blood, maybe ethical issues, regulation. What are some challenges that you're facing?
So the challenges are everywhere, of course. If it's the research, the biological, the funding, the regulatory. But the interesting things are, for example, the ethical question. There used to be a big controversy around the usage of embryonic stem cells, which we are not going to be using. We're using stem cells from people who gave the sample with their consent. So we're circumventing that. Also, I think most religious groups accept blood as fundamental for life saving. There's only one group which you may have heard of as Jehovah's Witnesses that are reluctant of receiving blood, and we're hoping that making an artificial process that is totally unconnected to the human body will allow them to accept blood also as a pharmaceutical product, an off-the-shelf product, just like a pain reliever. And perhaps we will be able to supply even for them red blood cells.
That's a very good positioning for the product. What about in our case, in India, what are some challenges you think we might run into?
I think the two things we need to consider. One is while developing all this alternative technology, one thing we also need to keep it is how much affordable it can be made. Because if you come up with a completely innovative technology, but if it is super expensive, adaptation might not be that good. So I think one need to consider to keep how we can keep this in a very affordable manner. And second would be it's a more practical operational one. Okay, so for example, when you have the alternate technology, different cell-based blood, it might not have much issue. But if you develop a new technology like a blood bag, for example, right? Your end user adoption, how much it has, because for example, your customers would be your blood banks and the clinicians, not your patient actually. So if you add any more process steps, operational steps into your existing process pipeline, often that might discourage them to put this additional effort to adopt the technology. More often, this is the challenging from the adoption. So that's why we are also very extensively, interacting with the blood banks and the clinic groups to see what is this entire operational pipeline, where without disrupting that, where we can seamlessly integrate this new part so that like it doesn't cost anything extra to them, and also it doesn't need to undergo the people who are operating a separate training or so on. I think these are the finally like a deal breaker. Sometimes it happens. So in fact, I just mentioned that this is a new blood bag, I said, right? But I'm still not sure whether it is going to be a bag or it is going to be a simple insert, which in the existing blood bag, we just put this insert that takes care of the cleaning of the blood. All these ones, I'm still yet to decide what is the final device, because in stuff like me saying that, okay, this is a great product, you take it. I want come that decision from the user and say like, in operational, this is the best design I can adopt. Then we can develop the technology in that way. I think thinking from the user point of view and custom developing for that will improve the chance of and accelerate the translational program. So that will be okay.
I've got one last question, and then I want to hear from you guys. My last question for the panel is, what kinds of partnerships are they looking for? What partners do they have already, and maybe what are they looking for in the audience over here? So, Ari.
Yeah, so we're looking for partnerships on the funding level, either from governments or from funds. We're looking for collaborations, pharmaceutical companies, bioproduction companies that are looking into producing blood for the population. So here in India, for example, and regulatory connections. So all of these, we need all of these connections to move forward. So
you're from a company, you're from a research lab, so I guess you're looking the other way, right? You're looking for companies to partner with you, you're looking for governments.
Government and companies.
Yeah. Praveen?
Yeah. So in fact, like we are planning to, as I mentioned, like we're already forming a spin-off company to take this to the clinical studies and so on. So anyway, we'll be raising the private equity money to do the conducting trials. But I think one immediate thing what we are looking is like we want to get this user data, as I mentioned, like interact with as many number of possible the blood banks and the clinicals to get the feedback from them about the final device designs. Okay, because and also there might be some sectors might need a specific design versus another sector. So I think we are at that stage where we can have more interactions to get exactly what market need is, what market wants, rather than what we want to give. So that's the stage we are.
Okay. Yeah. Okay, audience questions. You got about a little more than 5 minutes. I can see one hand at the back there. Can we get a mic to our friend over there, last table? Yeah. Just tell us who you are and which company you're with.
Hi, I'm Nidhi. I am from Exelor. I had an IP-based venture curation arm called Exelor Labs. I have looked at blood transfusion very closely, but from a different angle, and I wanted to get your thoughts on that. So the angle that we looked at is the testing. So in India specifically, we only do ELISA-based testing for most of the times. Most of the units of blood do not get NTA test. What happens as a result is we miss some instances where the blood unit that has been donated might be HIV positive. And as a result, what happens, what the data tells us is, for example, hemophilia patient in India who has to receive lot of frequent blood transfusions through the lifetime is 100 times more likely to contract HIV through blood transfusion as compared to their developed country peers. Right? And I wanted to get your thoughts on how the respective technologies that you are working on, how is it going to make those patients for whom blood transfusion is a lifeline, right? How is it going to make them safer from contracting infections like HIV, etc.? So,
okay, thanks. Good. That's one question. Any other questions? We can take a couple of questions back to back. Yeah. Let's have a question from here. There's a mic behind you.
Hi, I'm Rohan. So this is a scientific question. I want to understand, let's say, when you try to mass produce those RBCs, how do you plan to make sure that the RBCs that are differentiating from every stem cell are consistent? And also, to you, sir, this is a related question. So the blood that will be produced, right, from differentiating stem cells, what will be the life cycle, not life cycle per se, but the shelf life of that blood compared to the blood that's donated by volunteers? So those two questions.
Great. Any other questions? Good. Let's take these two. So one was about testing. How do you make sure that the test can screen out these other kinds of diseases? And the second question was about shelf life, consistency, and so on. So
great question, Nidhi. I think that this is where the regulatory body would make huge difference, because this minimum tests also that varies from sectors, and where you have the tier 1, tier 2, tier 3 cities goes, and this completely changes. I think there is a huge need, it's an immediate need where the regulatory bodies can have a uniformal guidelines to have these are the basic and compulsory tests they need to be done in order to pass through that. In fact, like many blood banks, they do, but like it's not uniform again. So, and once you have this contamination, I think it's a huge risk problem. I don't think right now there is any technology exists, like if it is already have an infection and so on. To eliminate that infection, I don't think we have any technology as of now. I think this can be prevented only by having the rigorous testing guidelines. That's the one thing. If other question you would like to take.
So actually, cultivated red blood cells will be totally safe for two reasons. First of all, you're manufacturing in a bioreactor, and the growth media that you use has no animal or human products, which means that you're not introducing any infectious carriers into your process. So when it comes to infectious diseases, cultivated red blood cells will be totally clean. On the other hand, another part of the side effects that are caused from blood transfusions are from residual blood components, not disease factors actually, but white blood cells, for example, or antibodies, other blood components that come with the transfusion or are not removed during the separation process. And also here, the cultivated red blood cells are differentiated into red blood cells, and in the production, you separate these cells. Only the red blood cells remain. Everything else is rinsed out. So on the safety side, they will be much more safe for infections and residual blood components. And of course, the blood type will be universal. So the mismatching that happens occasionally, not only because of the main blood type factors, but also those additional minor blood groups, will be eliminated because that we're using blood from what we call the universal donors. So all of these problems will be eliminated.
Wow, that's an amazing scenario. Yeah. The other question was about shelf life. How do you guarantee it's consistent and things of that sort?
Shelf life. So we're not testing that yet. We're still yet to move into scale, but there are already indications in the literature. So first of all, in our body, a red blood cell lives or works for about 120 days. From the moment a blood cell comes out of the bone marrow where it's produced, it has 120 days of circulation until the cell is recycled and the iron is reused. So when you take a blood transfusion, you have a distribution of cells that are either 1 day old and or 120. And that's why the quality of blood diminishes in storage, because all of the older cells start to decompose, start to get lesions, and lose their hemoglobin into the solution. That's one of the biggest problems in the quality of stored blood. When you produce the cells in a bioreactor, all of the cells are the same, are synchronized. and they're harvested at the same day. So even if one cell matured a day before or the day after, they're all harvested at once, and they start their 100 and some days from that point. So the starting point is already better. Adding them into better storage conditions will do that. And a clinical trial that's held in the UK has already shown that cultured red blood cells produced from progenitor cells, from stem cells, have a longer efficacy when injected to humans relative to donated blood. So there's also a good horizon in that direction.
Great. If there are no other questions, I have one last one for the panel, which is, you got a question? Yeah. That's fine. That's fine. Go ahead.
Thank you very much. Someone raises their hands for me. That's very nice. Thank you so much. My name is Peter Ling. I'm an IP lawyer from Switzerland. And just a very short question to Mr. Gargir, I guess, primarily. You mentioned the issue of stem cells and using non-embryonic stem cells. And it just made me think of this, of the regulatory and patent law framework in Europe, which, as you know very well, has been pretty bad in the last 10 years since, first of all, embryonic stem cells cannot be used or under very strict conditions, and the patentability of those inventions is restricted. And also last year, the European Parliament has also kind of restricted or proposed to restrict the patentability of some kinds of gene manipulated therapies. And my question, to be very short, is, do you see a future for research and development in Europe for this technology that we are talking about today? Or do you think that this will take place in other parts of the world?
Well, first of all, I believe that you will not have an alternative, that you will have to use similar technologies in order to solve the blood transfusion need. The population is growing, the elderly population is growing, the donor base is decreasing, and this is the way to solve it. The specific technology that we're using has patents behind it, but so you have license for using these technologies, the Yamanaka technology, for example, which has recently expired, but you could still receive the license for the production. We will not be doing, in our specific example, we're not going to be doing additional manipulations to the cells. So hopefully this will not consist of a problem. That's my view from a straw.
Maybe you can talk a bit later, but yeah, you want to wrap up?
Just to add that, I think there are two components, right? Like one is use of your differentiated cells for transfusion applications, and your other invention can be like having a different way of differentiating them to have a better quality RBCs, right? I think there's a lot many ways one can generate the IP from there. I mean, so even if you end use applications you protect, but still the process IP can be generated. There is a scope here. Yes.
Perhaps another point is that the source of the cells is on consent. And you know, there's a classical story about the Gila cells, and a lady that a cell sample was taken from her. She died back in the 50s, and her grandchildren sued Thermo Fisher for royalties on the production and many processes that were based on developments that were made on their grandmother's cells. So there was no consent, and there was no compensation. I'm taking that into consideration from the beginning, as definitely a person whose blood type will be able to save millions of lives should be compensated somehow.
Great, thanks. That's a great way on which to end this panel discussion, which is there are all kinds of amazing scenarios coming up, and we're just at the very early stages of what could be a very good chapter in the future of blood, the future of humanity, the future of life. So a big round of applause, please, for our two speakers, and you're most welcome to touch base with them again for further discussions after the session. But I guess we have another short coffee break now, but let's take a group photograph before we leave.
Thank you.
While you take the group photograph, you're absolutely right. We are doing a short break, and we will be reconvening in the ballroom at 5:40 for capitalizing on the innovation dynamics between startups and established corporations. While they take a picture, give them another round of applause, please.